Hello everyone, and welcome to today's The Scientist webinar. My name is Susan Harrison-Uy, Engagement Manager for The Scientist, and I will be moderating our discussion. Today, our panel of experts will discuss the analysis of complex biological networks to develop an integrated view of cellular systems. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the panelists will address these during the Q&A session following the presentations. To ask a question, simply type your query into the question box located on the bottom left-hand side of your screen. We will try to address as many of these questions as we can. However, if we do run out of time, I will forward any unanswered questions to the panelists and they can respond to you directly. The webinar platform is user-friendly. You can move or resize any of the windows by simply grabbing them at the top or stretching them at the bottom right-hand corner. You may need to move or minimize some of the windows to see the live view. The webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors. Bethel Laboratories Incorporated has been dedicated to supporting scientific discovery through its qualified antibody products and custom antibody services since its founding in 1972. Every antibody that Bethel sells has been manufactured to exacting standards at its sole location in Montgomery, Texas, and has been validated in-house by Bethel's team of scientists. Antibodies are tested across a range of applications, including Western blot, immunoprecipitation, immunohistochemistry, immunocytochemistry, CHIP, proximity ligation assay, and ELISA. Currently, Bethel's portfolio consists of over 7,150 catalog products, offering close to 5,700 primary antibodies, targeting over 2,700 proteins and 1,450 secondary antibodies raised against immunoglobulins from over 25 species. Trial sizes are now available and are conveniently priced at $50. To learn more about Bethel's products, please visit bethel.com. Rockland Immunochemicals Inc. supports the academic, biopharma, and diagnostic industries with antibodies and antibody-based tools used in basic research, assay development, and preclinical studies. With facilities in Pennsylvania for over 50 years, Rockland manufactures products ideally suited for integration into critical assays such as Western blotting, immunohistochemistry, immunofluorescence microscopy, ELISA, and flow cytometry. Additional information about Rockland's life science tools and services can be found on Rockland's website at www.rockland-inc.com. And with that, let me introduce our first speaker, Dr. Shannon McWeeney. Dr. McWeeney is a statistical geneticist working at the intersection of computer science, biostatistics, and genetics to develop approaches to solve research bottlenecks and novel ways to visualize and interpret information. In her work, her strengths have been in being able to think outside the box and her ability to rapidly synthesize information from diverse fields, which has allowed her to keep pace with rapidly evolving technology and target key areas early. An illustration of this is her substantial contributions with regard to genome-wide approaches for transcriptional regulations and chromatin occupancy. She was at the forefront of this area and a key part of the original team that developed the first scalable sequence-based approach, providing the computational workflows and methodology necessary for the approach to succeed. That work served as a springboard for a large number of other studies, leading to a tremendous increase in our understanding of transcriptional regulatory networks and cancer signaling, and serving as a precursor for next-generation approaches. Her work on novel computational methods and frameworks for prioritization is one of her most significant contributions to science. While these methods are designed for precision medicine, they have wide applicability for target identification and therapeutic prioritization for many complex traits. In 2010, she was selected as a Kavli Frontiers Fellow by the U.S. National Academy of Sciences for her contributions. Dr. McWeeney?
Thank you, and thank you to Susan and the Scientist for allowing me to speak. I'm honored to be part of this panel with Bing. I'm going to talk today about some of the integrated approaches that we've been utilizing to study complex traits. And to do that, I just want to give kind of a reminder about what we really are facing in terms of the disease spectrum. So we can think about this in terms of monogenic diseases. So these are the famous kind of poster children for this: cystic fibrosis, sickle cell anemia, and then the more complex diseases. And what I want you to notice in this figure is that we're really looking at potentially what is the genetic contribution versus environment. And this is obviously an oversimplification, but it's still important that in the monogenic diseases we're primarily focused on the role of the genes that are interacting. In complex, the environment becomes more important, still a major genetic component. And then we have a number of environmental diseases, and I'll mention a couple of these today as well, in which there's kind of a shift in that distribution, really a very heavy environmental component, especially when we think about things like exposure for infectious disease, but still a important genetic component that we want to be able to elucidate and really understand. We've also had this tremendous advent in our ability to measure and look at responses, and this can be when we're thinking about disease progression or when we're thinking about immune response in the use case of infectious disease. And so at the top part of the slide, what you're looking at are a number of the different modalities that have been developed, the different platforms that we have, which are ways that we can actually take measurements of these biological processes and these systems that we want to study. And I'm sure you're familiar with a number of these. We can think about, especially from sequencing, things like DNA sequencing, looking at genetic variants, transcriptomics, would be microarray and RNA-seq, and then clearly what has become much more important recently with a lot of the advances technologically is now population-level proteomics and metabolomics. All of these are providing complementary but also unique information. And what we are really doing now is trying to put this into a framework where we can understand across scales their contribution to these processes that we're trying to understand. Now, there's an important piece to this, which is really kind of maybe explain the focus of our work. We've seen this amazing evolution in terms of the way that we view the world and we think about science. And I'm referencing here the fourth paradigm, which was a speech and later a book in memory of Jim Gray, who was a scientist at Microsoft. And what he really summarized is this evolution that we've seen in terms of really our early work in just describing the world around us, classification, categorizing things, and then this important shift into the theoretical frameworks that really have been guiding us. We've just really emerged from this computational era. And we now have incredibly sophisticated algorithms and methods and data structures that can handle really a lot of the data that I just described to you that's being generated. And what that leaves us now with is this new era and kind of new challenge, the fourth paradigm, which is exploration. And that's how do we interpret this data? How do we visualize it? How do we find meaning and actually abstract information and knowledge from the data? And that's really where we're sitting now, and that's the focus of a lot of the work in my group and the work of others. And definitely I think you're going to hear that theme throughout today in the webinar. Now, people call this the era of big data, and the reference here on this slide is the idea of the data deluge. Really, we are generating a tremendous amount of information, and I want to remind you that it's not just the data types that I just mentioned to you, all these omics techniques, but we really are at the advent now of being able to think about all of these other information sources that are available to us. And on this slide you can kind of see a number of them, and some you're familiar with, although maybe not necessarily in a systems perspective and kind of the framework we're setting up for today, but they're critical. So if you think about something like infectious disease where you're interested in surveillance, and a number of these modalities come in when you're thinking about how are diseases transmitted, migrating, looking at things like air traffic patterns, GPS, satellites, also the ability for people through things like Fitbit and other systems to be able to monitor themselves, to collect personal data. In fact, all of this is really that, into that area. And what we want to be thinking about is how do we really maximize the information that we are now able to collect and to mine it, and to really help us in terms of these predictive models to really understand these systems. And this really allows us to now start to capture some of the information with respect to some of the environmental and exposure pieces that we were missing that are really going to be critical for us to understand this. The hope is from this that we will really be able to develop cutting-edge diagnostic diagnostics, therapies, and treatments. And this really gets into this concept of really having a systems-level perspective of disease. And what is critical to that is that there is a temporal component, or time, the importance of time. And so what you're looking at in this figure is 3 representations of gene networks. What is happening is we're looking at in the first 2 on the left, so at 2 weeks and at 12 weeks, there is no clinical manifestation of disease. But what I want you to notice is that we are definitely seeing changes that are starting to occur in the network that are representing disease progression. And only later, when we're actually manifesting, when you see the clinical manifestations, by that time there's already been substantial changes to the gene network, and this could be regulatory, this could be metabolic. There's a number of ways that you can think about where these perturbations are happening from this disease progression. It is critical for us when we think about things like early detection and intervention that we are able to develop methods that can maximize this information even when a patient is asymptomatic or there's no clinical signs, to be able to detect and to be able to target as early as possible. Now there's another component to this, which is that we also would like to be able, as a disease progresses, especially let's say they now manifest clinical symptoms and we assign therapy, we'd like to be able to continue monitoring this so that early, before we have a failure in terms of response to therapy, that we could make adjustments in their treatment plan as well. And I'll talk a little bit more about that. So what I want to do in my time with you is just briefly highlight 3 different kind of families of computational approaches that we've been working on in this kind of systems framework, thinking about both predictive models and prioritization as well as discovery, and each one is important. And I'm going to narrate them as a series of use cases. So when we start with some of our network-based methods, what I want to focus on first is infectious disease. And so we're thinking here about, in particular, respiratory diseases and respiratory disease signatures. And I'm highlighting a couple of things that have made the news in the distant past and also more recently. So one of the major pandemics that we saw in terms of respiratory infection was obviously the 1918 influenza pandemic. And then more recently we've seen the SARS pandemic in 2002 and then SARS-like outbreaks in 2012 and more recently again with MERS and SARS-like viruses. This is a continued issue, and so we're thinking here about what are the strategies that we can utilize, especially coming from these network methods and system methods, to be able to identify potential signatures for respiratory disease. Now, a common model for this is the mouse model, and I'm just showing you an illustration of one of the types of experiments that we can do, in which we are looking at the top the idea of animals that are infected with the virus, and this could be SARS or influenza or other viruses, and we can look at different doses. And then we also have animals that are basically mock infected, where there's no presence of the virus, but they still go through the same type of infection process, just so we can control for any confounding factors from the actual means of infection. And then we can look at, and this is really important in the animal models, we can actually look at some of the key sites. So in this case, because it's a respiratory virus, we're interested in what's happening in the lung. And we can look across time to look at changes in the network or in the system. And in this example that I'm showing you, which was work that we had published earlier, we were looking at both gene expression and at proteomics. And so from this, what we want to be able to do is to develop an understanding of what is happening in terms of the network. And I'm showing you an example just for the proteomic data, but it's important to understand that we do this for all of the data types. So we can do this for proteomics or for expression. What you're seeing at the top, the circles are these ideas of modules. And so what we're thinking about, the different color coding, so notice blue, orange, pink, these are representing aggregate signatures of genes. They're highly correlated, and this is a discovery context. You'll notice the word de novo. So what we're interested in is not being biased by our existing knowledge, but using the data in front of us without any pre-existing bias of known pathways or known function, to really ask what is the underlying structure. And we're able to look at the changes. Remember, this is a time course, and we also had a change in terms of the perturbation of both dose and infection status. And so we can ask, how are these changes being manifested? How does this change the underlying network structure in terms of the correlation structure and the relationships among these genes? And it is these subnetworks or these modules that are actually very interesting because they can represent potential signatures. And the reason we're interested in that is because we can actually correlate those with various phenotypes. So for example, we can correlate that with clinical phenotypes such as in the example of infection, weight loss, or lung pathology, etc. Can you see the screen right now? I'm showing nothing on my screen. I just want to make sure that everyone can see this.
Yes, we are still seeing it, Shannon.
Okay, great. So it's just gone blank on my side, so forgive me.
Your browser?
Yeah, okay, no problem. So I'll keep going. I just wanted to make sure. I was worried I was talking to. So are you on this? Can you see the slide that says network?
Proteomic networks.
Yeah, no, I've actually moved to the next slide. So let me. So you want me to refresh my browser? Let's see. Hopefully this won't kill everything. All right, hold tight. We're rebooting. It says that there's an error while trying to retrieve the event.
I can advance the slides for you. Do you have a copy of your
presentation that would be great.
Available?
It is showing. Yeah, you'll have to tell me what slide you're on because, oh wait, no, it's rebooting. Here it comes. Apologies. Hold on. Let's see where we are. All right. Tell me if you do you see now the slide that says network biomarkers? And if you don't, Susan, I'll just have you push for me because it's not showing anything on my end.
Yes, I do see it, but I'm not seeing you online at the moment, so you may actually be offline. So I can advance the slides for you if that will help.
Okay, that'd be great. Let's just do that way so we can keep, because I don't want to take over Bing's time. So if we're on the slide that is entitled Network Biomarkers right now, hopefully. So what we want to focus on here is this idea that we have from these subnetworks or these modules, we have these aggregate signatures, and we can actually ask how correlated they are with different biological phenotypes when we think about the mouse model. But when we move, importantly, into patients, we can ask how well they're correlated with clinical phenotypes. And so that's a really critical aspect of this, is to be able to, from a de novo perspective, to be able to identify novel signatures that actually might be biologically or clinically meaningful from these networks. If we move to the next slide on network integration, what you can see here is one of the challenges. So you notice I showed you a network from proteomics data. We're collecting multiple data types, and so one of the things we really have to think about in a very serious way, and this is a major challenge across the field for systems biology, is how do we integrate these different data types. So I've already mentioned how they're important in terms of their ability to give us different nuances but also to be complementary. And so we really have to think about how we can bring these data types together and really find novel ways to integrate them. And that's one of the things that we were focusing on here, were different strategies where because we don't expect there to be, and this is very important, a one-to-one correspondence between the data types. We know that's true in terms of both the timing of when you see changes, for example, in protein versus expression, just using that example, or more importantly, that there may be modifications or other things that would not allow for that one-to-one correspondence. But we do want to have a meaningful way that we can start to integrate them. And I'm highlighting here just a couple of ways that we were thinking about that. This is an open question. This is one where there's a tremendous amount of work. And what we were really trying to lay out and evaluate were these different ideas of either kind of early integration prior to building the networks, or late integration where potentially you've built the networks and then trying to bring them together, and to make this as meaningful as possible in terms of new discovery, where the data types together are really informing either what is the critical underlying structure of the network, and more importantly, where are the potential signatures or biomarkers that we might be able to utilize. Now I want to move to my next slide and talk a little bit about another use case where the use of network methods really comes in, and this is a motivation personally, for many of us I say almost all of us at the Cancer Center where I work at the Knight Cancer Institute, we really are starting our timeline when we think about when a patient comes in at diagnosis. And one of the critical pieces for us is to be able to determine what the right therapy is for that patient, what is the right dose, what is the right timing, and to be able to do that really on an individualized level. And so if we go to the next slide, I'm highlighting here a workflow in terms of precision medicine that we utilize at the Cancer Center in terms of really following the patient through. And one of the things that I want you to notice is that at diagnosis, when a patient comes in, we do a tremendous amount of characterization of the samples that we receive from that patient. And there's two things that we're looking for. We're doing much of the omic characterization that I've already talked to you about, so looking at changes in expression or looking at changes in phosphorylation or changes particularly in variation, so things like mutation. We also do a series of functional assays, and the reason that we do this, and I'll show you this in a minute, is because we want to integrate this information to actually help us in terms of prioritization. And so this is key. Besides just development of signatures, which I discussed in the infectious disease case, we really want to be able to use some of these network methods to prioritize therapy. And you'll notice it's a cycle, because what's going to happen is that patient may or may not respond to the therapy. And depending on what happens, especially because we have, in a way, this kind of evolutionary arms race between the changes in the cancer and in the mutational profile in response to treatment, we need to be able to be ready with the next combination or knowing what the right combinations are to target the specific cancer that that patient has. So if we go to the next slide, you'll see an illustration of work that we had developed in terms of really utilizing some of these network methods to guide prioritization. And what I want you to notice about the color coding is, so this is data from a single patient, and so this is done per patient. And what we're looking at is, and the network is just a visualization of this, but what we've been asking is, are the different, and the circles, remember, are the nodes or the genes in the network, and we're looking to see, based on the color coding, if these were functional hits from either the siRNA or these small molecule screens that we do. We're looking to see if there's evidence of overexpression or mutation, and we take all of this information and we prioritize. We have algorithms that we've developed that will actually rank and prioritize so that we can really look at what appears to be the critical mutations for this patient, and then from that determining if they're actionable and guiding our clinicians and our oncologists in terms of the prioritization of therapy. So if we go to the next slide, what this really hints at is that this has really started to reshape the way that we think about clinical medicine. And we now, there's always been this sense of kind of team medicine, but really we're at the interface now of team medicine and team science, really system science, in terms of thinking about how to bring in all of these computational methods, all of these advances in terms of the platform, and really make sure that we can integrate them and really have a systems medicine approach. So if we go to the next slide in terms of the discovery component, so the last example that I want to discuss in terms of the methods approach is work that we've been doing where we're really thinking about how do we start to use some of these network approaches to really understand the underlying disease etiology and to start to tease that apart. And so I have an example on the next slide in which we're highlighting some work that we've done in which we're really trying to understand about gene-gene or polygenic interactions and associations. And this is actually quite critical in a disease sense, and I'll just briefly highlight that. When we think about a number of the large-scale cohort studies that we do, often these are studies in which they're looking gene by gene, so marker by marker, and looking for any significant association. And what we end up is in a scenario of what we call main effects, where they're looking for, is there any strong association from a particular gene that would indicate an association with the disease or the phenotype being studied. What we are often missing is the interactions among genes, and so these may not be significant main effects, but there could be these secondary interactions that actually are just as important in the disease. And in fact, we know for many complex traits, we have a lot of genes that are contributing much smaller effects, but actually contributing significantly to the disease, and we really need to be able to understand how they are interacting together. And so we've started to utilize different, again, network-based methods. I think it's really pulling a lot of what we know from machine learning and other fields to start to try and detect these associations. And so if you go to the next slide, you can see an example of where we can begin to map these gene-gene interactions, these polygenic associations, basically epistasis, into known pathways and networks where we can really begin to ask questions about the role of this as well, not just single gene contributions, but really started to get at that interplay and novel ways to detect them. And I'm going to mention to you that the critical point here is, are we clear of the ways that we can detect these smaller associations when there is no, in the absence of any significant main effect? So if we move on to the next slide, we're really focusing on just a couple of brief comments about where we are on the horizon. And the slide after that, I think one of the main things I'd like to stress with you is that we really are just beginning to tap into the potential from the electronic health record. So this is a really critical source for us. There are a number of funded studies that are going on. One of the most famous is the eMERGE Consortium, in which they really started to look at how do we start to pull this information in to use it for better phenotyping. And this is a critical piece of this. I've focused a lot on the genes, the interactions, the proteins, but we really need to understand about the actual phenotyping that's involved as well, and potentially from that cohort development for better studies. So that's a very active area. And if we go to the next slide, we also have a key piece as well in terms of really starting to bring in much more of the information that we have in terms of exposure. So some of the chemical etiologies when we're thinking about the environmental diseases and toxicology, and also really beginning to better utilize these network methods. And there's some fantastic work being done on this to really highlight at chemical and drug interactions, drug-gene interactions, and really start to map that out. So those are two areas where I think there's really exciting potential in terms of bringing this information in. We've been developing a lot of the methodology. When I say we, I don't mean myself alone, but I mean many groups have really been active in this area to really now have the tools where we can really start to think about how do we ask these questions. So in terms of just briefly some next steps, I think it's really clear that we have a number of ways that we can be thinking about this in terms of how to really take this information and make it actionable. And I think one of the things that I stressed early on and I want to just end with is that we really are trying to make the data as accessible as possible. And again, I don't mean can I download the data. I mean, can I interpret it? Can I extract information? Can I extract knowledge? Can I put my hands around it and really explore it? And that's really the major challenge that we are all facing and contributing to. On the next slide, I just want to briefly mention some of my contributors from my group and collaborators who are key for a lot of the methodology that I've shown you. And then on the final slide, just want to highlight the partners and funders who really have made all of this work possible. And with that, I'd like to hand this back over to our moderator, Susan.
Thank you very much, Dr. McWeeney. If you're able to see if you can get back online with your system while Dr. Zhang is going through his presentation. Thank you. We have a lot of great questions coming in. Please continue to submit your comments, and we will address them in the joint Q&A session after the next presentation. Now I'd like to welcome Dr. Bing Zhang. Dr. Zhang is an Associate Professor of Biomedical Informatics with a joint appointment in cancer biology at the Vanderbilt University School of Medicine. He received his PhD degree in molecular genetics from the Chinese Academy of Sciences in 1999. His postdoctoral research at the University of Tennessee, Knoxville, and Oak Ridge National Laboratory focused on bioinformatics and functional genomics. He joined the faculty at Vanderbilt University in 2006. Dr. Zhang has more than 10 years' experience in the area of bioinformatics, proteomics, and systems biology, with more than 60 publications in these areas, including papers published in high-profile journals such as Nature, Nature Methods, and Molecular Systems Biology. Bioinformatics tools developed in his group include WebGestalt, NetGestalt, CustomProDB, among others. WebGestalt is one of the most widely used pathway analysis tools with more than 1,000 cumulative citations and more than 1,000 weekly visits. Dr. Zhang's ongoing research focuses on the development of integrative bioinformatic approaches that help translate experimental data into biological and clinical insights. He currently serves as principal investigator, bioinformatics core director, or co-investigator on multiple federal grants. Dr. Zhang serves frequently as program committee member in international conferences and reviewer for multiple NIH panels. He also serves on the editorial board of multiple journals, including Molecular and Cellular Proteomics and Clinical Proteomics. Dr. Zhang?
Yes. So good afternoon, everyone. So can you hear me?
Yes, you sound great. Thank you.
Yeah, okay. So first, I would like to thank Susan and the scientist for giving me the opportunity. And it's a great pleasure to do the presentation with Shannon, who just gave a very exciting presentation on the high-level overview of systems medicine and with several very good examples. So my presentation will be more focused on a specific area based on our recent work on making sense of network hairballs. So this is closely related to the fourth paradigm just pointed out by Shannon, the data exploration and visualization. And also, I think important part of this work from this presentation is to try to make big data accessible to ordinary biologists and clinicians. So as you already seen from the previous presentation, high throughput technologies have basically revolutionized molecular medicine. And for a disease sample, now we can easily do genome-wide measurements at different molecular levels, at DNA levels, RNA levels, and protein levels, right? And molecular alterations at these different molecular levels eventually exert their effect through altering protein and network activities. So protein-protein interaction network has become a very good model for visualizing, integrating, and analyzing different types of data. And the last decade of systems biology studies have shown that complex diseases are the result of dysregulation of networks instead of individual genes. So networks are modeled as graphs and visualized as node-link diagrams. So, for example, this is a small protein-protein interaction network, and each node represents a protein, and the two proteins connect to each other if they interact with each other. And so you can see the connection between the molecules of proteins. And interestingly, you can also overlay experimental and annotation data to the network. For example, you can highlight a few nodes based on specific annotations by changing their shape to triangles. And then you can also overlay the color or change the shape to represent different type of experimental measurements. For example, you can use red color to indicate genes that are upregulated and green color representing genes that are downregulated. I think you have already seen several beautiful node-link diagrams from Shannon's presentation. And these type of diagrams have been frequently used in publications. So this is great, but when we go to the whole protein-protein interaction network, for example, this human protein-protein interaction network with almost 10,000 proteins and 36,000 interactions, and the node-link diagrams become a hairball map, right? It's difficult to get any detailed information of this hairball map. And moreover, if we want to overlay different types of data, for example, the copy number alteration, mutation, methylation, or mRNA expression, splicing, protein expression, modification, overlaying these different types of data onto the network, even for a small network, it becomes very difficult. So one thing we're trying to do here is to try to come up with a new method that can help visualizing multiple types of omics data together on a complex network. So try to make this scalable. And our approach is based on trying to make sense of the hellboard. So this is still the protein-protein interaction network. Although it looks very messy, and the complex network studies have shown that most of the complex networks, including the protein-protein interaction networks and other biological networks, usually have a hierarchical modular architecture, meaning the proteins first connect to each other to form small networks, and then growing into bigger ones, and eventually most of all the proteins are connected to each other, right? And now, if we use a horizontal bar to represent this whole network, and we can see that this horizontal bar can be separated into 4 smaller bars, each represent 4 of the small subnetworks. For example, the one I highlighted here is represented as the first short bar, right? And then each of these short bars can be further divided into 4 smaller bars, each representing a smaller module within the network. And finally, we can put the network nodes under the modules corresponding based on their relationship. And now, although originally in the node link diagrams, we used both dimensions, the horizontal dimension and vertical dimension, to lay out the network nodes. Now we only use one dimension to lay out the network nodes. Although we won't be able to see the detailed interaction relationship between the nodes, but the hierarchical modular organization of the network is very well maintained because this layout is derived from the network structure. The beauty of this transformation is that now we can lay out other types of data as tracks in the vertical dimension. So data can be visualized as tracks. So if you used genome browsers before, this is very similar to the concept of genome browsers, using tracks to visualize different type of data. But instead of organizing the elements based on the genome sequence, now we organize the genes based on their relationship in the protein-protein interaction network. And of course, if we look at this small toy network, it's easy to see the hierarchical modular architecture. But when we go back to this big network, right, the protein-protein interaction network, it's difficult. It's difficult for me even to convince you there is such a structure. So that's why we have developed a method and called NetSam, trying to reveal the hierarchical modular architecture of complex networks. What we do here is to start from a protein-protein interaction network, and then we can calculate all the pairwise distance between these nodes. For example, we can use the shortest path distance between each node pairs, right, to represent the distance on the network. And of course, we can use more sophisticated method to build the distance matrix. And after you get the distance matrix, you can do a hierarchical clustering based on the matrix in order to get a hierarchical tree, like the one shown on the left side under the network. But the problem of a hierarchical tree is that all the branches can be actually switched, right? The same tree, as shown in this two smaller hierarchical the dendrograms, they are the same hierarchical trees, but then the node can be organized in different ways. But what we did in this algorithm is to try to optimize leaf node ordering to minimize the sum of pairwise distance of the adjacent nodes. And the next thing is, from a hierarchical tree, it's difficult to tell where are the modules, right? And what we did here is to try to identify the best partition and do statistical evaluation to test whether there is hierarchical modular architecture of the network. And then if there is statistically significant structure, we make the partition. For example, we can cut here as indicated by the red bar, and then we get 2 smaller modules. And then we can repeat these steps and keep cutting until we cannot find any significant modules. And after that, so basically, for this small toy network, we can find 3 hierarchical levels and multiple modules at different levels. So this tool is, the NetSem is implemented as an R package, and it's available in Bioconductor. And now you can apply the same method to the complex network, like the protein-protein interaction network, to convert the 2-dimensional hairball into a 1-dimensional clear layout of the proteins in the network. And we also showed that for the human protein interaction network, we identified 4 hierarchical levels and 92 modules. And we showed that most of these modules have high functional homogeneity or spatial homogeneity or dynamic homogeneity. So basically, these are not only structurally identifiable, but they correspond to biological units. And now, after we convert the 2-dimensional layout to 1-dimensional layout, the genes are ordered in the horizontal dimension, and now we can visualize data as tracks along the vertical dimension. So we use a barcode plot to visualize single binary tracks or significant genes or gene ontology annotations. For example, we can each of these black bars represent a gene annotated, let's say, to be cell cycle, and then you will be easily you can easily identify where are the cell cycle genes located on the 1-dimensionally ordered network nodes. And we use the heat map to visualize composite binary tracks. So for example, this visualizes the mutation status of many samples. The samples are showing as the rows and the genes showing in the columns. And we can see at the bottom, there are many samples. Around 10% of these samples have a lot of mutations. This may correspond to the hypermutated samples. And then we can use single continuous track to visualize the fold change or p-values for individual genes if you do an experiment. And then we use the heat map, again, to visualize composite continuous track, for example, gene expression matrices. And in this example, we can see on the left part of the screenshot, you can see under the green bar, these genes are basically all upregulated at the samples at the bottom group. And we also implemented integrative data visualization and analysis tools in the system, and this system is called NetGestalt, and the system has already been published 2 years ago. So, and last year, in order to demonstrate how we can use this system to help biologists to access the large amount of omics data, so we built a NetGestalt-based colorectal cancer portal. So we collected data from colon cancer the tissues as well as cell lines. And for both tissue and cell line samples, we have data at different dimensions, like mutation, copy number alteration, methylation, mRNA, and protein expression, etc. And then all these data are available from the same portal, and the users can easily retrieve and visualize and analyze the data. And this portal is also described in detail in a paper just published. And next, I will give you a quick demo. And this is the website, NetGestalt, which can be accessed at www.netgestalt.org. And if you go to the colorectal cancer portal, you will be able to access all the data and tools as shown in the next demo.
In this video, we are going to show an analysis of the frequently mutated genes in colorectal cancer, or CRC, using the NetGestalt CRC portal. This is the user interface, and this is the track viewing area. The default view is derived from a human protein-protein interaction network based on the IREF database. The view can be changed through the View menu, and the data tracks can be added to the track viewing area through the Track menu. There are multiple methods for adding tracks, and we will just browse the system tracks. Tracks in the system are organized in a Tree menu, and we can open all branches using the Expand All feature. Among the different types of available data, we are interested in somatic mutations in CRC. There are several tracks under this category. We can get detailed information on individual tracks by clicking on the names. Let's add the mutation profile track to the viewing area. Let's also add the pre-computed list of significantly mutated genes. The mutation profile track visualizes the mutation status for individual genes in individual samples, with genes on the horizontal dimension and samples on the vertical dimension. Some samples may have many mutated genes as indicated by the red. These samples may correspond to the hypermutated tumors. To show the hypermutation status of the samples, we can open the drop-down menu, available from the track title. There are several analysis features available, and the subtrack annotation feature allows us to visualize various sample annotations. Let's add the hypermutation annotation. Now all the hypermutated samples are grouped together. You can reorganize the samples based on any of the available sample annotation features. Let's close the subtrack annotation panel for now. The track titles can be hidden to get a better view of the tracks. In the track of significantly mutated genes, the genes are not randomly distributed on the network. To test whether these genes are significantly enriched in certain network modules, we can use the network analysis feature available from the drop-down menu. There are several network analysis options, and we will perform the module enrichment analysis. The analysis identified 4 enriched modules. We can add these modules as individual tracks by clicking on their names, or use the Add All Modules feature to add them simultaneously as a new track. In this new track, the pink boxes highlight the enriched module, with mutated genes colored in red. Let's zoom into one of the modules. After zooming in, the names of the genes are automatically displayed. Mutated genes in the module include BRAF, NRAS, KRAS, and PIK3CA. We can visualize the node link diagram for genes in this module, with mutated genes highlighted in green. Network modules enriched with mutated genes are likely to be biologically important. To better understand the module, we can add the omics snapshot track that summarizes different types of molecular alterations, which include somatic mutation, somatic copy number alteration, epigenetic silencing, and differential expression at mRNA and protein levels, respectively. Now we have a comprehensive view of all molecular alterations associated with this mutation-enriched module. This concludes tutorial.
So this example shows how we can overlay data to protein-protein interaction network and co-visualize different types of data under the same network. But as Shannon already mentioned in her presentation, this type of approach is kind of limited by existing knowledge, right? How about if the interaction relationship is unknown, then you will not be able to see that. So in addition to mapping data to a knowledge-derived network, it's also possible to build networks from the data directly. And this slide shows an example we just recently published on the gene co-expression network analysis. So basically, from the gene expression data, we can build co-expression networks. So in these networks, each node is a gene, and 2 genes are connected by an edge if they co-express with each other in multiple tumor samples. And in this figure, the figure B, and this actually shows the co-expression network, and the different colors highlighted a few modules, the big modules. And what we did next was to filter these modules using data from the mouse models of metastasis. So basically, we ask, are there any of these modules are enriched with genes that are upregulated in the genes that we see upregulated in the metastatic mouse model compared to the parental cell line? And as you can see in the GSEA analysis, we identified one of these modules with a lot of genes that are upregulated in the metastatic mouse model compared to the parental mouse model. And then we can ask in this module, what is the common regulatory mechanism for this module, right? And then by doing the transcription factor binding site enrichment analysis, we identified unfed, a transcription factor that might be the upstream regulator of this module. And then the shRNA knockdown or pharmacological inhibition of NFAT-C1, we showed that the target genes, identified target genes of NFAT, are actually downregulated by the perturbations. And we also showed that this gene signature, including the genes regulated by NFAT, are actually able to separate tumor patient, colon cancer patient, into those with good prognosis and bad prognosis. And we also show that in the mouse model, altering NFAT activity can actually change the metastatic potential of the cell lines. So in that collaboration study, so basically my group did all the computational analysis, and then we generated the hypothesis, and my collaborator's group actually did all the experimental validations. But then our collaborators asked us, it looks very cool, to do all this computational analysis, but can we do this by ourselves, or will you be able to teach us to do this by ourselves? So after we have the NetGestalt framework, we try to see whether we can implement this into the system so that the biologists can also use this type of tool to do the analysis by themselves. So this is not available in the public version yet, but in our development version, what we can do is to upload the gene expression data into the NetGestalt system, and then the co-expression network will be constructed, and then the co-expression modules, just like from the protein-protein interaction network, and similarly from the co-expression network, we can also identify the co-expression modules, and you can also the heat map actually shows the data that helps generate this co-expression network, and you can already see that for the modules, let's say the module under around 1,000, so the genes under the same module show very high similar expression pattern. And then you can do the statistical test to ask, for individual gene, which of these genes are associated with patient outcome or recurrence-free survival, and then we can this result can be visualized as another track, and we can see the module on around 6,000, this region, there are a lot of genes that are positively correlated with increase the risk. So we can also do the analysis at the module level that can help us. So by considering each module as a metagene, and this identified a few modules that are either correlated with increased recurrence risk or reduced the recurrence risk. And now we can also visualize the mouse data, and we can see under one of the red modules, the genes are actually upregulated in the metastatic mouse model compared to the parent model, and the pattern is very obvious. You can also co-visualize the NFAT targets and then do the enrichment analysis, and we show that the NFAT targets are actually also enriched in this region. So by performing this simple analysis in NetGestalt, you will be able to link your human data to the mouse data and to existing knowledge on the transcription factor. So that can basically help you to generate hypotheses as we did before for that paper. And then you can also zoom in to this region, this module that are likely to be related to the increased risk of recurrence, and then we can see all the recurrent as labeled by the green on the left. All the recurrent tumors have higher expression of genes in this module, but for the non-recurrent ones, a lot of them have very blue color, right? Meaning the genes are only expressed, but also some of the non-recurrent ones also have red color. So that means for the currently non-recurrent tumors, maybe this module can be used to predict the future recurrence of the tumor. And you can also see the mouse data at the third track. And when you go to this level, you can also visualize the module the in node-link diagrams, and you can covariate the mouse data with the human data, and you can also covariate the human data with the transcription factor target information. So this can help you to quickly generate potential targets for follow-up experiments. So to summarize today's presentation, we introduced NetSem, a tool for revealing hierarchical modular organization of complex networks, and NetGestalt is a web-based tool for integrating multidimensional omics data over biological networks. And the NetGestalt colorectal cancer portal allow you to explore different type of data from colorectal cancer genomic and proteomic projects. And we recently added the co-expression network analysis in NetGestalt. And for future work, we are going to build data portals for other tumor types, and we will implement features so that users can upload data to generate networks, and this is not currently available. We can also consider other hierarchical and linear organization of genes, and this can be potentially applied to other domains. And actually, we just recently tried to apply this to a single-cell data analysis and have shown some interesting results for that type of analysis. And finally, I want to thank people in my group who built this tool and generated all this data. And also, I thank the funding source for that supported the work. And thank you all for listening to this presentation.
Thank you, Dr. Zhang. Quite a few questions have come in, so we'll move on to our Q&A session. Please continue to submit your questions or comments via the question box. Dr. McWeeney, how are de novo protein networks analyzed? Would unsupervised cluster analysis be an appropriate method?
So I'm going to back up. So for a de novo technique, and that doesn't have to be a proteomic network, it could be expression. You have to decide, one, what you're going to use basically is the information source. And so the methods that I was showing are all based on correlation. They are distinct from clustering, although there is a overlap a little bit in terms of some of the module detection, or, which overlaps with that. But strict clustering, one of the problems with it, as people who are familiar with this know, is that you're always guaranteed to get a cluster, and it becomes very difficult to so if you take data, any data, good data, bad data, put it through a method, you're going to get clusters out. And so one of the things that we want to do, number one, is be able to assess any type of clusters or modules that we're detecting, how robust they are. And so what I didn't go into because of time is that there are a number of methods that we utilize to actually ask how distinct or how robust the modules that we're detecting are. And some of these actually do bring back in some of the known information, but we don't rely on those alone. So one of the things you can ask is how well supported are these clusters by known protein-protein interactions or other things. That's actually very limited. And one of the things we found out early on is that the clusters themselves, the modules, actually are often poorly annotated, which is actually what you want. You want to be able to find things that we didn't know about before, and it turns out that right away you're going to start to discover subnetworks or modules that don't have a tremendous amount of annotation. So we do look at things, so this is really based on this idea of a transformation that's happening to the data, in which we're really looking at the what we call the topology of the network. And so one of the things that we do assess immediately is to make sure that those modules or subnetworks are actually distinct from just random topological networks. And that's a really critical phase of that. So long answer short, because I think I'm rambling and I apologize, you could use a discovery method like clustering. What you need to be careful about is that number one, you're going to get an answer and you need to be able to assess it. And number two, depending on the data that you're using, the method, the algorithm, et cetera, they'll you'll probably have a lot of issues with stability, and this has been shown over and over again, especially in clustering methods of expression data. So the approach that we were showing is actually a bit distinct from that, and a lot of it is the heavy lifting up front of how we're actually processing and really looking at what we call topological overlap. And this is because one of the central metrics that we're interested in is connectivity and really thinking about centrality. So how important is a gene to that modular network?
Thank you. I'll pass the next question over to Dr. Zhang. Are there identifiable characteristics of networks that would reliably indicate whether they stabilize or destabilize in the aftermath of perturbations?
I think that question will rely on probably time course data, right? If you would be able to build a network based, there are two things. One thing is I'm not quite sure whether the question is about us mapping the data to existing network or building network from experimental data. I think for the mapping data to the existing network, if you can have a time course experiment, for example, and then you can do the module enrichment analysis or something like that for at different time point, and potentially you will be able to identify the modules that are altering in the predefined network that may stable if there is actually a stable state of that. For the constructing of network, I think that is probably a different question. The question then is whether you get the same network structure, right, as the perturbation going on. I think it's still possible, but that will probably require a lot more data in order to make that analysis.
Thank you. Dr. McWeeney, what do you think the main challenges of integrating different types of omics data into systems-level analysis to generate clinically actionable biosignatures?
Great question. A couple, and I'll try not to write a book in my answer, but I think one of them is the timing, and this is really important. I showed a little bit about that in one of my overview slides about how we recognize that there is a tremendous amount that's happening from a systems perspective way before we see any indication clinically, especially in terms of manifestation of phenotype. What is essential is that we are actually sampling correctly when we think about this. So if you think about the examples that I showed you with proteomics and expression, just to take two, and of course if we're looking at multiple measurements, this would obviously increase in complexity, but let's just take those two. One of the questions is, are we actually sampling correctly when we're taking these samples to really capture what is happening? And the problem, as you can probably imagine, is that if we aren't sampling on a fine enough scale or aren't sampling during the critical windows at which we're changing, when we're seeing changes in the system, then we would miss it. And so you can really think about this from something like, let's say, ChIP-seq, which is interested in looking at chromatin occupancy, so transcription factor binding, and expression. If we're very interested in looking at changes in occupancy, where we would then expect to potentially see changes in the expression program, if we are not actually capturing the correct time points, then we will have an instant issue of potentially thinking that nothing has happened, when in fact we may have missed that window. And so that's one of the first things that we face is, are we sampling correctly, just in terms of the timing that we're after, because this is definitely a systems level question, and we have to think not only about the different data types, but about what are the critical points or junctures for that. As I discussed as well, we also have these issues in terms of when we're trying to integrate the data. There are a lot of things that we didn't go into in the presentation that both Bing and I face in terms of how we process this data, how we're mapping it. So with proteomics, for example, we're actually measuring peptides. And so we have an identification or mapping issue when we think about how we are reassembling those into proteins. We have issues in terms of protein translation, so post-translational modifications that have actually changed the message, where we potentially have a lot of additional error that is coming in. And that's before we even get to the really exciting thing, which is then to say, how in my model do I integrate those? So those are a number of uncertainties that we really have to grapple with and kind of face head on and be realistic about. And that is why it's so critical not just to be utilizing these types of systems approaches, but also to really stress the idea of reproducibility and having lots of people doing these experiments, making sure that we can actually reproduce the results, and trying to generate as much information as possible, and also as many conditions. And Bing just kind of discussed that a little bit with the perturbation. So we need to understand that we actually have captured enough information to understand how the system is changing. And what's very important to understand is these changes may not manifest in the measurement we're making. So for example, we've seen a number of times where we can see differences in the network. So the connectivity of the network, the relationship among the genes will shift, but yet there's no differences in or change in expression. So if you did something like differential expression analysis where you asked, between these two conditions, do I see anything changing? No, we don't see anything, but yet you can see substantial changes in the network. And so that tells us that there are additional readouts that we need to be capturing that we either are not doing, either because we're not using the appropriate techniques, or again, the sampling and timing issue.
Thank you. Dr. Zhang, we had a question come in for you on can the disease outcome be decided based on the protein-protein network in the infectious disease between pathogen and host?
Well, I think that's a very interesting question. And basically, you will have to be able to build an interaction network, not only including the host side, but also the pathogen side, right? But I think several groups have already started that type of work. So if you can have a protein-protein interaction network with proteins from both sources, and then if you have also measurement from both the pathogen and the host, and the data can be mapped to those networks. But remember that, and then you can either identify the modules or identify the critical part of the network, or even certain types of network characteristics that are associated with the clinical outcomes. But I just want to emphasize that this type of analysis are likely to be just association-based. So all these analysis will probably require perturbation-based studies, hypothesis-driven studies, in order to actually figure out the causal relationship, because most of the networks we are using are still interaction-based network or association-based networks, and also the relationship between the network features and the outcome is also based on the association type of study. I think it's very useful to help you to generate hypotheses, but the causal relationship will require testing hypotheses generated from these exploratory studies.
Thank you. Another question for you, Dr. Zhang. Somebody asked how the networks were built. Were they based on correlation matrix showing their relationships?
So I talked about two types of networks. The first one is the protein-protein interaction network. That is knowledge-driven. Basically, you can go to the protein-protein interaction databases and collect all the pairwise interaction relationship already defined, either based on high-throughput experiments or based on existing publications. And then for every pair of proteins that interact with each other, you can make an edge, right? And then you will be able to build a protein-protein interaction network from that. But for the second part of my talk, I was talking about the co-expression network. So that is based on experimental data. In the example I used, it's based on gene expression data, either from mRNA microarray or from the RNA-seq, right? Let's say in this example, you have measurement for, let's say, 20,000 genes, and 100 tumor samples. And then for each gene, each pair of genes, you can calculate their correlation based on the expression pattern across 100 tumors. And then if they are have similar expression pattern across the 100 tumors, then you make an edge between them. This way you get a co-expression network. Of course, this is just one very simple way to build a co-expression network. There are more advanced ways to build a network based on a model-based approach or rank-based approach instead of just based on the correlation scores. But the general idea is to make connections for the genes that have similar expression pattern. And this can be extended to other type of experimental measurements. For example, protein expression measurements can also be used, right? And if you're interested in metabolomic studies, and then you have metabolite measurements, then you can also build a metabolite co-expression network. Yeah.
Thank you. Dr. McWeeney, can these networks be stimulated to reveal network dynamics?
Oh, absolutely. So these are, it's hard for both of us, I think, to really communicate that in a presentation, but this is very much a dynamical system that we are studying. And so the perturbations that, the experimental perturbations that Bing talked about, yeah, there is this idea that we can perturb the systems in order to elucidate dynamics robustness, for example, something we're very interested in. What we have to be careful about is the combinatorial aspect of this, because even if you take a very simple, and I say simple in quotes, but very small sub-networks that you want to study, when you start to bring in perturbations, because this is connected, because there are, and remember, some of this are things that we're discovering through like the de novo methods that we've both talked about, and some of these are things that we know. And even the interaction backbone, the protein-protein interaction, our existing knowledge that Bing just discussed, that is, of course, based on specific conditions and time points. And so it's very possible that, and we know this is true, that these interactions can be changed or modified. And so yes, it is entirely possible. That's one of the things that we actively want to do. In fact, when we think about systems biology, when we think about systems medicine, there is this idea not only that it's temporal and that we have this aspect of time, but that we are able to look at how the system changes dynamically in response to changes. And this could be environmental exposure, this could be changes in the system, these could be controlled perturbations. That's critical. And the other context that we didn't really get into yet is also, especially when we're thinking about human systems, changes just in response to where that system is in its own timeline. And so when we think about, for example, an infectious disease, we see dramatic differences when we look at the immune response in our modeling the immune system in terms of infants or children, pediatric cohorts versus the elderly, versus adults, and that's a reflection of not just the genetics in the system, but also and the perturbations via a pathogen or a virus, but also other exposures. So absolutely we can do that, but it does become very complex very quickly, and that's often why you'll see some of the perturbations that we've described or are kind of alluding to in terms of single gene knockouts or very controlled things where we're really trying to just understand and map how that kind of resonates through the network.
Thank you. Dr. Zhang, we had a question come in about the software that you described. Is this available? Where could somebody find the software if they wanted to use it?
The mapping data to protein-protein interaction network is publicly available. So you can either use the network that already exists in the system, or you can also upload your own network and mapping your data to the network. But constructing network, de novo network, as Shannon also mentioned, for especially the co-expression correlation-based network, we are still working on that part to try to implement that into the system. But because this is a web-based system, and constructing a network is kind of time-consuming, so we are trying to first try to make it faster, and secondly, try to build some queuing systems so that the job can be queued, because you won't be able to build a network in, let's say, 1 minute. and then that will require a slightly different system that can handle the more time-consuming jobs for web-based tools. But if you already have a network and you want to map your data to the network, or if you are only interested in the protein-protein interaction network we already have in the system, then you should be able to do that very easily.
Thank you. I'll pass the next question to Dr. McWeeney. How much uptake has there been for a systems biology approach to complex non-cancer disease areas such as immune-mediated arthritis or asthma?
So I'll try to partition that because I kind of highlighted for the cancer use case one in which we were really thinking about prioritization of therapy, and for many reasons, cancer is kind of one of the obvious places to start there, especially given what we know about the cancer heterogeneity and the need for that. Now, that doesn't mean that that's exclusively in the domain of that, and I would reiterate that a number of these approaches are utilized across the board in complex diseases. So the interesting one that was brought up is obviously anything in terms of autoimmune or immune-modulated diseases where there's a really tremendous component of both genetic variability and then obviously environmental components as well that come in depending on which one we're looking at. So these approaches are being actively utilized, especially when we think, and remember I kind of broke it down into a couple of different things, prediction of signatures, prioritization, I was giving the therapy example, and then discovery. And I would say that across the board in complex traits, you're seeing more and more an integration of these types of systems approaches and integration of these methods in all of those areas outside of cancer, really very heavily in the predictive component and in the discovery, and starting to see it more in the prioritization, although it really does necessitate that you have kind of a framework for that already set up, especially clinically or in terms of a clinical research or clinical trial framework. But definitely they are, and I should comment that, one of the things that I touched on with infectious disease and immunology, so there's massive funding in systems biology coming from NIH, really focused on assessing the feasibility of these approaches, really thinking about understanding immune response in general, and that leads itself to really a number of things, not just infectious disease, but also when we think about both the role of the immune system in any complex disease and also things like immunotherapy. So kind of across the board, we're definitely seeing this being more integrated and utilized just at varying levels. And it also depends really on the stage, as I mentioned, of where for that particular scenario people are in terms of the clinical studies that could correlate this, especially when you're thinking about prioritization for therapy.
Thank you. I'll open up the next comment to both of the speakers. Somebody asked for your opinions on a Sidney Brenner quote, which is low input, high throughput, no output. What are your thoughts on this kind of system analysis? Do you see it as moving quickly towards clinical applicability, or do you have any thoughts on this?
So could you repeat that question? Sorry.
Sure. The quote was low input, high throughput, no output. So it's a quote that comes from the thought of factory science, where you're creating a whole lot of data and struggling to analyze it and figure out what is useful. Are you seeing that you're able to get clinically relevant information through your analysis?
So let me.
Go ahead, Bing. I'm sorry.
Okay.
No, I was just going to take a different standpoint on it, and maybe I'll talk quickly because I want Bing to be able to answer it as well. When I think about that, I really think about the fact that often we're not really sitting down and really asking about the questions and the direction that we want to move in. And I think this is the critical piece, and that kind of gets at some of the low input part. So we really need to be having a very critical conversation about what is our end goal, what does success look like. And this is very important when you think about a number of the efforts that are happening right now across the spectrum in basic science, translational science, and clinical research. So if we have a clear idea of what we are trying to get out, then we can better direct the experiments that we're doing and really not just end up with a lot of data and struggling to make sense of it, but really have very specific questions. Now, there is totally a role for discovery in this, and there have been a number of compendium and large-scale resource studies that are really critical for expanding our knowledge of what we really understand, and that data is being utilized over and over again in multiple approaches and is valuable. But we really do, to me, I would emphasize that first piece. It is essential that we are really thinking about what our question is, making sure that when we are doing these studies, we have thought about what are the right time points, do we have the right samples or subjects, are they well phenotyped, so that we can actually ensure that at the end we are seeing something that is meaningful and that is headed in the direction that we're trying to go.
So I want to mention that although we're all talking about big data, but in my view, I think the limitation is still that the data is still small. And the reason I'm saying this is because, although we are able to make measurements, for individual samples, right, we will be able to measure different molecular molecules at different levels. For example, you can, before you, let's say for tumor, you only have a dozen clinical attributes, right? But now you can have maybe millions of measurements for individual sample. But the problem is, although we have a lot of data for individual samples, but you only have very limited sample numbers for any type of large-scale studies. So no matter how many measurements you can make from individual samples, that doesn't give you better chance to do discovery, without increase the sample size. So I think the sample size is still critical, although, we can have a lot of data, but if the sample size is limited, then I think the discovery is limited a lot by that dimension. So a lot of people come to me, and when they try to design experiments, for example, they want to do 1 versus 1 or even 3 versus 3 experiments, but they try to measure all types of, from RNA, DNA, protein. But I think if you are limited by resource, by money, I would rather measure, put the money, for example, only on proteomics, but do the study for more samples. I think you have to balance the both dimensions, the sample size and what are you going to measure for individual samples. So if you only pursue one dimension to make that very big, and then you don't have money to get enough samples on the other dimension, I think your study will be very limited.
Thank you. We had a question come in about if either of you could recommend good training opportunities for basic biology researchers to be educated in systems biology approaches, including software and statistics applications.
So there's a number of them, and I would also make a call out at this point to initiatives that NIH is doing right now. So we, along with several others, are funded through the Big Data to Knowledge initiative, and there's 2 components to that I really want to draw attention to. So one is open educational resources, and these are materials that will be freely available online and are available, really covering a wide array of training geared at different levels. So depending on where someone's coming from in terms of the gaps or the skills that they're trying to address about how do I manage or analyze or even think about this data, how do I pose the questions, how do I begin to use some of these approaches. So all of that material is starting to come online and will be available over the next few years. And is available at no cost. And then as well, there are a number of workshop and skills courses that are being offered. We will have one, and ours are being targeted, and different institutions vary, but are at different levels of learner. And so depending on that individual, and people are happy, of course, we're happy to have them email us, but I can talk more offline about that. But there are a number of opportunities through the Big Data to Knowledge, or what we call BD2K initiative, and that would be one place to look. And then there are outstanding short courses and workshops, as well as, obviously, depending on how serious someone wanted to do in terms of their training, certificate programs, masters, obviously the other OER or MOOCs online courses. So there's a number of opportunities, and I think, Susan, if you'd like, I could provide a list for you maybe that the scientists could post related to the webinar if people are interested.
Yeah, that would be great. Unfortunately, we just have time for one more question. And so we had a question come in about how you can technically integrate the different types of omics data that were generated by different platforms and have different units, such as reads, area under the curve, etc. Do you have any tips on that?
Yeah.
Who's the question to? Maybe Bing could answer that one because I just spoke. Or I'm happy to help if he needs.
So I think the challenge, even just for molecular data, is the heterogeneity of the data, right? For example, the measurements are, for example, in the NetGestalt tool, we simplified by, mapping all types of data to the gene level. But actually, for the phosphoproteomics data, you actually have data at the phosphosites, and then you have data at the peptide level, or at the, I think depending on what you want to find, what is your final question, you have to decide what is the unit that you map all different types of data to, if that is necessary, and then you can go from there. I think it's very context dependent. Shannon.
Yeah, no, I would completely agree. That's exactly what I was going to say. It's like he and I are reading each other's minds. I think it is exactly that. It's context dependent. What's the question you're asking, and what is that unit? And this is very important when you think about the network approach you're trying to employ, because certain ones are going to kind of constrain you to a gene or protein or other level, and so that's going to be very key. One of the things that I would strongly suggest is, if you can, to actually look at the data so you really understand. For example, if I'm using a microarray, how are the probes designed, and what am I actually measuring? Versus if I'm doing RNA-seq, where I might be getting not just one transcript, but all of the transcripts possible, depending on my depth and coverage. So completely agree that it is going to be context dependent, and you really have to be thinking about the approach you want to use, the question you're asking, and also what is the data that you have generated actually measuring, because that's going to really lead a lot of the mapping strategy.
Thank you. Unfortunately, that's all the time we have for today. If we didn't get to your question, I will forward them to the speakers and they can respond to you via email. If you have any further questions, please consider reaching out to the speakers directly. Their email addresses are displayed on the screen. As a reminder, the webinar will be archived on the Scientist website, and you will receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today and for sharing your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Dr. McWeeney and Dr. Zhang, as well as our webinar sponsors, Bethel Laboratories and Rockland Immunochemicals. Thank you, everyone, and goodbye.